because I like to learn things too. And so it's a real thrill to now be here in front of you all. I'm going to talk about this question: can living cells assemble themselves? And we'll start out with a thought experiment. Suppose you have a complex machine, like a computer, and you immerse it in a wearing blender, and you wait until it's ground up into little tiny granules, and then take it out of the wearing blender. What will happen to those granules? I've never done the experiment, but I can tell you, I think for a fact, that nothing will happen to those granules. They will never again do computations. The computer is dead at that point. All right, so how about if you take a different complex machine, take some frog eggs and put it in the blender, and then take it out of the blender. Is it ever going to function again like a frog egg? I don't know. That's actually the question that we're going to address here today. And there's a guy in my lab who addressed exactly this question by accident. And he discovered something really interesting. So this is the guy, Sean-Ray Chung, and he was doing a research project that was sort of the typical kind of thing that all of us do in science. We had some hypothesis that we thought was probably true or maybe true, and he designed some experiments that would allow him to test that hypothesis. And I'm going to run through that as sort of background here. A hypothesis was about how cell death spreads through cells. In my lab, we've been very interested in how things make their way from one part of the cell to another part of the cell, which can be pretty trivial in small cells, but in big cells, it's a more interesting question because you've got cells like a frog egg is a millimeter in diameter. Your biggest muscle cells are 30 centimeters long, one cell. Your longest neurons are a meter or two long. So it's an interesting question. How does information get through these big cells? It can't get through by protein diffusion, because protein diffusion is just way too slow for that. So that's something we've been interested in. And Sean-Ray set out to see how cell death, that's sort of two of the big things that cells are born through cell division, and cells die. And that's it. Sean-Ray wanted to see how cell death spreads through cytoplasm. And he had a hypothesis. The hypothesis was that cell death spreads as what's called a constant velocity trigger wave. So let me start out by asking what is a trigger wave, and why is it that we thought that cell death might spread as a trigger wave. We have to know a little bit about how cell death happens. It happens in all sorts of different ways in cells. But this is the most organized and best understood way that cells die. It's through a process called apoptosis. Apoptosis was named in the 1960s after the Greek word for falling leaves from a tree, sort of a poetic way of thinking of your cells dying. And the way apoptosis works is somewhere in the cell there's damage, and the damage is bad enough that the cell thinks that it should die. One kind of damage is that the mitochondria, which are the energy storehouses of a cell, leak some of their contents out into the cytoplasm. And those leaked contents, this stuff cytochrome C, can cause this enzyme, caspase, to become activated. And when caspase is activated, it's a protease that chews up other proteins, and that's how it brings about death of the cell, by chewing up proteins. So cytochrome C appears somewhere in a cell as a result of damage, brings about caspase activation. Now suppose you've got a cell that's shaped like a long tube, just to make things simple. And suppose you've got some damage at this end of the tube, and cytochrome C has leaked down and caspase is activated here. What will then happen? Well, a fraction of these cas-based molecules will diffuse this far, and then a fraction of those molecules will diffuse this far, and a fraction of those will diffuse that far. And pretty soon you've got really tiny amounts of cas-base out this far, and you've got less impetus for the next cas-based molecules to move even further. So you might expect it would be hard for that end of the cell to die in a coordinated fashion with this end of the cell. But, as it turns out, one of the things that active cas-base does is it causes the thing that made it get activated. It causes mitochondria to leak out more cytochrome C. So you've got a positive feedback loop. A little bit of active cas-base can bring about the generation of more active cas-base. So if a few of these molecules make it here, that can bring about the release of cytochrome C from this region and activate up the cas-base to make it just as dead as this part was. And that can march on and on and on and on. And you'll end up with a wave of cell death that never peters out and never slows down. And that's what a trigger wave is. Trigger waves are familiar in biology to those of you who know neurobiology. Action potentials are trigger waves. They go at constant speed down an axon in a nerve cell. Calcium waves in tissues like brain tissues are trigger waves. So they're familiar in different areas of biology. They're also familiar in different areas of real life too. A fire spreading through a field, that's a trigger wave. None of the hot molecules from this part of the fire make their way across the field, but they make their way locally and cause this bit of grass to catch on fire, and then those molecules cause this bit of grass to catch on fire, and so on. So it's the same kind of deal as this. The spread of a fire is a trigger wave. The spread of a disease through a population is a trigger wave. The spread of a favorable allele through a population is a trigger wave. The spread of a meme on the internet is a trigger wave. So this is a class of phenomena that is familiar in biology in all sorts of contexts, but also in non-biological real life. The key for trigger waves is you need positive feedback plus some mechanism for local spread. And you really don't need anything else. All right, so Sean-Ray Chung thought that apoptosis probably spread through cytoplasm by a trigger wave. And the way he tested that was by taking cytoplasm, which can do apoptosis in vitro, and putting it in a really long thin tube. By really long, I mean like maybe 8 millimeters long. At a distance of 8 millimeters, it's really easy to distinguish a trigger wave that marches up and up and up and up from just diffusive spread that slows down and peters out. So if you put cytoplasm in here and put a bunch of nuclei in the cytoplasm, because nuclei disappear during apoptosis. And then he stuck this tube of cytoplasm into a reservoir of apoptotic cytoplasm. And the thought was that apoptotic mediators would diffuse their way up into this tube, and then we could see, does apoptosis march its way up the tube, or does it peter out? I'll show you the movie, and you can decide for yourself. It looks, kind of linear to me. And you can check to see if your eyes are fooling you by just plotting the position of a nucleus and the time at which the nucleus is disappearing due to apoptosis. And it's a nice straight line, and you can go long distances and nothing ever peters out. So Sean-Ray built on this observation to do some mechanistic studies and perturb the positive feedback loops and so on, and published himself a nice paper. And normally after publishing a nice paper like that, you'd go on and do the next apoptosis thing, but Sean-Ray didn't, because he noticed something here. Let's look at this experiment again. here at the start of the experiment, these nuclei that we put into the extract are kind of randomly dispersed through the extract. We haven't placed them anywhere, they're just wherever they might end up. But look what happens to where they're positioned by the end of the experiment. They organize themselves, or they get organized into these clumps. At the top of the tube, it's clumps of 3 nuclei per clump. Down at the bottom, there's some that are 1 and some that are more than 3. It doesn't seem like it's exactly a constant number of nuclei, but the spacing is pretty regular.
Is that real time, by the way?
This is sped up. So to go from the bottom of the tube to the top of the tube takes about 100 minutes. And by the way, any of you who have other questions, please feel free to ask. I like being interrupted from my normal flow. I like hearing you talk better than I like hearing myself talk. All right, so does this self-organization have anything to do with the fact that that cytoplasm was doing apoptosis? Well, there's a simple experiment for that. Put the cytoplasm in the tube, but don't stick the tube in an apoptotic cytoplasm. And look at this. They do the same thing. They regularly space themselves out. So Chandray thought that this would be an interesting phenomenon to investigate. How best to investigate it? Well, these tubes are great for figuring out that it's a trigger wave, but a round Teflon tube is not the ideal kind of thing to do microscopy on, as it messes up the optics. Really what you want to do is something kind of more normal. Just have some cytoplasm sitting between a couple of cover slips. And turns out it's important that you coat the cover slips with something like Teflon, a really inert kind of surface. But once you do that, you can do this experiment. This is going to show you a sped up movie. This will be about, I think, 45 minutes total. This is just cytoplasm sitting between a couple of cover slips. And you can see it starts out just looking like a bunch of disorganized garbage. And then that happens. The cytoplasm organizes itself into what, for the life of me, looks like a sheet of cells. So, see, these kind of lighter things that are in the middle of these sort of darker islands, those are nuclei. And you can see some of the islands have a bunch of nuclei. That one has 2. This one has 1. This one I think has 1, although I think it's out of focus. And in between the islands, there are sort of boundaries that are lighter in color. So,
were the cell walls of the original cells ground up in that cytoplasm too, or was it just somehow cytoplasm only? I mean, sounds like a dumb question, but...
No, it's a good question. I'll tell you. These were frog eggs. They weren't actually put in a blender. They were put in a centrifuge tube, and we got rid of all the extracellular buffer that we could, so it was almost solid frog eggs. And we just spun them really hard in a centrifuge at about 100,000 times gravity. And at that speed, the shear stress on the cells will make them kind of burst and squeeze their guts out, squeeze their frog juice out. We end up with 3 fractions. The bottom fraction is mostly pigment granules that make the frog eggs the color they are. And the top fraction is mostly lipid storage vesicles that the frog embryo uses as food as it's developing. And then the stuff in between is murky, mocha-colored. It contains cytosol, which is the liquid part of the cell, plus endoplasmic reticulum, which is a membrane-bound part of the cell that is used for protein processing, and mitochondria, which generate energy, and lots of other like, little bits of plasma membrane. So there's all sorts of stuff in the cytoplasm, but it's disorganized at the start. All right, so what are all those things in there? One way you can tell is by doing the same sort of microscopy experiment by adding fluorescent markers for various components of the cell. So here's this green stuff is fluorescently labeled tubulin. That's one of the main structural proteins of cells. This is a dye that'll stain the endoplasmic reticulum, which is one of the main organelles. And this is a dye that'll stain the nuclei. And we can watch what happens to each of those things here. All right, so this is kind of like the movie that you saw originally. You're seeing nuclei ending up in the middle, these dark islands, these white boundaries in between the islands. The dark islands consist of the microtubules and all the organelles. This is endoplasmic reticulum, but the mitochondria are sort of similarly distributed. And then the gaps between the dark islands seem to be mainly just the liquid portion of cells. They don't seem to contain cytoskeletal elements or organelles of any sort. And then the nuclei end up in the middle. All right, so how to get some sort of insight into how this process happens. One thing you can do is just watch the movie and see what's the first thing you see happening. And to me, the first thing that I can see happening is organization of the tubulin. It seems to come from these three spots and to kind of blossom out from those spots in a star-like structure that's actually called an aster because it looks like a star. And then develop from that aster into this more wreath-like shape. And it turns out those points where the asters are, those come from the reconstituted nuclei that are in the extract. Reconstituted nuclei come from sperms, and sperms have microtubule organizing centers on them. So our strong hypothesis was, got a bunch of sperms in here, they've got microtubule organizing centers, that allows the microtubules to get organized, and then slightly later, all the vesicles end up being kind of hung on the scaffold that the microtubules are providing.
You said these are eggs, right?
These are eggs, frog eggs, yeah.
So what do you mean by the sperm?
To make nuclei, we took frog sperm and put them into the frog cytoplasm.
Okay.
And they turn themselves from sperms into nuclei.
Okay.
It's sort of magical. All right, so an experiment you could do would be just leave the sperm out. And I think we'd all expect probably no organization would happen, but here you go. So there's no nuclei at the end of it all. And there's no hole in the middle of the organelles for the nucleus to sit in at the end of it all. The microtubules look about the same. They take a little longer to organize, but they still organize. All right, so what is required? Let's see, microtubule polymerization is required. You can put into these extracts cancer drugs that prevent the microtubules from polymerizing, and they absolutely block this organization. You need organelles. The liquid part of cytoplasm won't do this. You need this motor protein, which is able to move organelles along microtubules. So that's all kind of holding together. You need energy. If you deplete energy, this won't happen. And you need to not dilute the extract very much. And this might be why people haven't noticed this before. Usually when you lyse cells, you have maybe 1 volume of cells and 100 volumes of buffer and you put them in the blender. With frog eggs, you can lyse the frog eggs in centrifuge without diluting the contents very much. If you dilute more than about 40 or 50%, the extracts lose their ability to do this self-organization. What's not required, you don't need to make proteins, you don't need microtubule organizing centers, and you don't need the other big cytoskeletal protein actin to be even present during this polymerized state. I'll show you one more thing. These look like cells, sorta. Can they do things that cells normally do? One thing that extracts, frog egg extracts will do that is again a miracle, is they'll do the biochemical version of the cell division cycle. So the cell division cycle takes place when you synthesize this protein cyclin, it turns on a regulatory protein, and then the cyclin gets degraded, and then it does it again and again and again. And every time the cyclin goes up and then immediately down, the cell divides. In these extracts, we can make it so that the cyclin goes up and down, up and down, up and down. So the question then is, what happens to these structures if the thing that drives the cell cycle is going through essentially cell divisions? Will these structures divide? And here's the movie. Here's the dark part is like where organelles are. The light part is like a boundary region, and red in this case is nuclei. All right. So these things not only look like cells, they'll divide like cells and produce daughter structures that are smaller replicas of the mother structure. So in summary, I showed you that apoptosis spreads through cytoplasm via what we call trigger waves, and trigger waves are these things that are like rumors spreading or forest fires. Frog cytoplasm, if you hold it in interphase of the cell cycle, will organize into these cell-like units. And if you let the cell cycle proceed, these units divide into daughters that reproduce the structures of the mothers, only on a smaller scale. So what does this mean to us? Well, frog eggs are different from computers. Maybe one of the fundamental properties of the components of cells is their ability to self-organize. And we kind of knew going into this that biological stuff self-organizes, but it does to a greater extent than we had suspected here. So maybe a basic property of living matter that distinguishes it from non-living matter is its ability to self-organize at various distance scales. So I'll end with that and show you again Chandra Chung, who did all the work I'm showing you tonight, and thank you.
So mathematically speaking, what is the trigger wave? What is the trigger wave, mathematically speaking? So, what, some kind of equation?
Trigger waves happen in mathematical models. A trigger wave is a self-regenerating constant velocity wave that depends on positive feedback. I showed you, have you ever seen the Belousov-Jabotinsky reaction? Those are trigger waves.
So I noticed you got rid of the lipid droplets in the beginning.
Yeah.
And they may have contained the components that help make a cell membrane.
Right.
So the question is, do your reorganizing units put the proteins that would be cell membrane proteins at the boundary between the cells?
This is